Well, thank you for the opportunity to join today's session. I'd like to discuss preclinical and vivo. I'd like to start with my email address in case you have any questions during this virtual format. Please feel free to send me an email and I'll follow up. Here's some research collaborations that I'm involved with, conflicts of interest. But perhaps most importantly, here's the outline for today's presentation. And we'll be discussing how to incorporate image analysis into the study plan, issues of data quality, image registration, and then a brief introduction to rigor in imaging studies, just some general thoughts. And in fact, I should begin by also pointing out that there is a fair amount of scientific management in this presentation today, in addition to just image analysis, which tends to be a rather dry subject. So I thought I'd kind of a little bit. So this first topic of incorporating image analysis into the experimental design. Well, why is it important? And here is a great example, as happens to be from our laboratory, where we perform CEST MRI studies, in this case of triple negative breast cancer patients. And we can perform what is known as an MTR asymmetry analysis or Lorentzian line shape analysis to produce these CEST maps that are shown here. And I won't get into the details of CEST, but we do acquire these spectra shown in red, or kind of an inverse of that. Here is the MTR asymmetry spectrum, showing that there is some kind of contrast at about 3.5 ppm that might be interesting for evaluating these patients. Well, I can do different saturation powers in the acquisition for these patients, and we've tried two different power levels of 2 microtesla and zero point 0.9 microtesla. And you can see that we get different quality of data, and the smoother data acquired at 2 microtesla is actually analyzed quite well using this MTR asymmetry analysis, comparing one side of the spectrum with the other. It's a little more noisy with a lower saturation power because we are comparing the noisy data on the right side with the left side to do this asymmetry analysis. Or we can do Lorentzian line shape analysis. We can fit the spectrum to a dip, find that peak, and then say that that peak is representative of the CEST contrast. And at zero point 0.9 microtesla, we actually get to see a narrower dip in the spectrum than at 2 microtesla, where the dip is there, but it's much more broad and more difficult to see. So right away, you can see that the acquisition method does affect the choice of the analysis method, which is also shown here, where if you're going to do MTR, I'm sorry, 2 microtesla for a saturation time, while MTR asymmetry does produce higher CEST values, does seem indicative of the decrease in the patient protein level from baseline to cycle 2 and cycle 4 of treatment. For comparison, if you're going to use the zero point 0.9 microtesla saturation power during your acquisition, well, then the analysis should be done with this Lorentzian line shape method, which produces better results, seeing a decrease in this protein content in the tumor with respect to therapy. So again, you have to ensure that the acquisition and the analysis method are complementary to each other. And I'm sure many of us have seen cases where the analysis is performed, or acquisition is performed, and we have a stack of images, and then we start to think about the analysis and realize that the acquisition could have been performed in a slightly better way to be complementary to the analysis. So yes, analysis has to be part of the study plan up front. And in fact, this is my personal paradigm for setting up a study. We have a rather business-like approach at MD Anderson. And so we like to create a study plan, which is really a draft of the methods of the pending manuscript. And it can be quite detailed. We could even have some introductory information in the study plan that could be the precursor to the introduction of a paper as well. The study plan can start off in a very draft form and incomplete, but it's circulated to all participants so that everybody agrees before starting that the study plan is full and complete, and acquisition and analysis and all the other parts of the project mesh well together. There's also an evergreen document, that is to say, we review these documents every 2 to 3 weeks to make sure that we're on course. So any project that involves more than one person really should have a study plan, in my opinion. So again, the sections of a typical study plan are to have a purpose, which could be brief, a timeline, very important to define exactly what day zero is in your timeline. I've seen some misunderstandings there. A section about materials, and then image acquisition parameters. And one would think that you could just list See all the image acquisition parameters in previous publication, or just use our standard method, whatever that standard method might be. But I think that's a mistake, and it's really important to list all the acquisition parameters that you would normally see in a well written scientific publication, because ultimately you're going to publish the results, so it's good to have that up front. But then also, then the analysis sometimes depends on those acquisition parameters, and it's good to have that documented. As I'll show on the next slide, I think it's really important to also think about data transfer and archival. All the information about the study should be together, including the analysis software. Many of us use MATLAB to analyze imaging data. MATLAB M files are very small, kilobytes in size. Why not store your MATLAB M file with every single data set? I know that takes time, but if you ever want to go back to that data set and know which M file you use to produce those final results from your analysis, well, it's nice to have that M file there. Otherwise, you might be scrambling thinking, okay, which version of M file, which MATLAB processing routine was used to produce these results? And that can be really challenging. So we like to store everything we can within the data files that come from our Bruker machine. Image processing is typically just drawing ROIs. I call that mouse radiology. And I think it's an important step prior to getting to the analysis. And very often in the studies that I'm involved with, we have one mouse radiologist drawing the ROIs, and then another researcher who's doing the advanced analysis with MATLAB, who may be less familiar with where all the organs are in the mice. So it's good to consider those two parts separately. And then finally, you should also consider your deliverables, especially if this is a service project. So just to emphasize that point again, here is the imaging pipeline that I would use as a paradigm. I do a lot of contrast agent development, so there's some chemical and biochemical tests that might occur in vitro, in vivo development of a model. And then finally, the acquisition. Now we often think that this is the pipeline. The job is done as soon as we acquire the images. And then the analysis part, well, that's pretty easy. Someone will do that work, right? And you've heard that story before. Somebody magically will do the analysis. Well, if you think about it, the analysis involves this data transfer, which is sometimes not trivial. Also archival, making sure you can go back to the data when you need it is non-trivial. And then image processing, again, is this idea of drawing ROIs or some initial tidying up of the data, curating the data, and then finally, the image analysis, which we normally think of as the true analysis part, but really it's a component of this whole pipeline. And then finally, the deliverables. You can analyze data, but unless you put it into a meaningful format for others to understand that information, your analysis is really limited in its value. So you really do have to consider the deliverables as part of this pipeline. Now, you could say this pipeline is not exactly to scale, that the top level here is actually quite long and the lower level is quite short. But I would actually disagree that this lower part of this pipeline, which I drew intentionally as its own row, is actually really important to consider. And again, here are some notes just to show why the data or the details are needed. For example, are you going to include lab notes, a list of what happened during your scans? The mouse died, the mouse did not have good respiratory gating, the mouse temperature fluctuated too much, for example, which is not captured in the imaged data. Plans for analysis results and analysis software should be archived as well, as I already mentioned. Also, the deliverables. What are you really going to give to the end user? And that might be a publication. You could think of the manuscript you're writing as your end user as well. And so here are some examples of the data set or data files that we actually write down in the study plan that this is the goal. Like begin with the end in mind. And then here's the important point on this whole slide and why the second row of the pipeline is really highlighted as its own row. I really feel that for each hour of acquisition time, you got to plan for 2 to 3 hours of analysis time. Archival, transfer, processing, analysis deliverables. It really adds up. And there are many of us who plan a study, calculate how much scanner time we need, how many dollars we need to buy mice and cells and so on. And we completely forget about that analysis time. But look at the labor involved. It is huge. And we really have to consider analysis as a major part of this pipeline if you're really going to do studies correctly. I'd like to switch topics and talk about image data quality. Here's one example from DCMRI to show that you really have to think of what is the quality of your data, your acquisition, and how will that affect your analysis procedure. And I really think we sometimes need to think outside the box and not just think incrementally about data analysis. And here's one example. So, as many of us know, tumors have rapidly growing vasculature that's very chaotic in nature, very tortuous, and very leaky. And so, when a contrast agent is injected into intravenously into the mouse, the contrast agent can flow or perfuse into the tumor through the endothelial wall of the vasculature and collect in the tumor. And you can see that we have bright contrast in a flank tumor here after injecting a standard gadolinium-based agent. The bladder also gets bright. I like to point out that I've been with many graduate students who have seen this image for the first time with their experimental agent. They get very excited to see the bladder light up, and then I need to remind them that that means 98% of their efforts have gone to the bladder and not to the flank tumor, but that's how physiology works. So, a flank tumor after 24 hours of an anti-angiogenic treatment was also studied, in this case, a mouse. And you can see a necrotic hole and less uptake of the contrast agent in the periphery can be observed. And so we're all familiar with how DCMRI can monitor changes in angiogenesis within a tumor. Um, but what we don't like to acknowledge, although we're all familiar with it, is that this arterial input function, which is needed for comparison to the pharmacokinetics of the agent in the tumor, is actually quite noisy. And here's a renal arterial input function. Circling a tiny dot near the moving kidneys is not a fun exercise to do. You can lock down the legs a little bit better. I prefer that to get the femoral AIF. But still, there's motion there as well, and that little dot is very challenging to find in some legs. The pharmacokinetic profile of uptake and decrease after injecting a gadolinium-based agent in the tumor is really not too bad, but there's some noise there as well. So here's my first question. Why are we taking poor quality data, the AIF, and trying to make good DCMRI results through the analysis? To me, that doesn't make a lot of sense. Now, let's start with good quality data, not with bad quality data. Well, there's no way to get better quality AIF, so instead, what I prefer to do is what Tom Yankelov and also Greg Karsmar have shown, and using the reference region model, where the pharmacokinetic uptake in muscle can be compared to tumor rather than just comparing a capillary to a tumor. And I think you get better results in doing that because it's easier to find a muscle and you have better quality data with the muscle. So this can be fit using a nonlinear least squares reference region model. It is challenging because now there are two pharmacokinetic curves, one from the blood vessel to tumor and blood vessel to muscle that have to be compared. So there's significant noise by comparing those two data sets. This is shown here in kind of a less than average example, quite noisy for the tumor and muscle data that's here. And you know the fitting is weighted towards regions where the data is changing rapidly. When you do nonlinear fitting, you need initial guesses. I haven't heard anybody say that they really enjoy nonlinear data analysis within MATLAB. It tends to be slow, sensitive to noise. So, you know, you can prove this situation with better hardware and temporal sampling, but we've linearized that model. Here's a publication about that. And then you can apply some smoothing methods, and you almost get a straight line. There is some systematic error to this, but it's nearly linear, and therefore you can do linear data fitting. Fitting is equally weighted throughout those points, independent of initial guesses, right? When you use Excel to fit a straight line, you don't have to give an initial guess. And the cumulative average is less sensitive to noise. And again, you can apply some smoothing as well, which helps. So now we can avoid the noisy AIF, and also the noisy tumor and muscle data can also be mitigated by using analysis methods to our advantage. So it's good to think about the quality of your data and then find the analysis method appropriate. And is the linear reference region model helpful? Well, yes, it's about the same as if you were to do the nonlinear reference region model with good initial guesses. If you use a standard guess, one value for all pixels, well, the repeatability is not as good by this gauge repeatability and reproducibility metric. And details are in this paper by Jones et al. And you can even see that the linear reference region model map looks smoother than the nonlinear reference region model map of relative k-trans. By the way, no guarantee that the linear reference region model map is correct. There's not a gold standard here, but it does have this intuitive feeling that it should be smoother. To this slide, just to show that again, there are ripples of some kind of data or information in these spectra in this data that's difficult for you and I to see with our eyes, but machine learning can pick out some features, while the DCMRI is a bit noisy and even difficult for the machine to pick out those features. And T1 relaxation, just not enough information there for even the machine to see a difference between the models. So it's interesting to think that there are cases of effect size where there is some information but higher than the noise in the data that even if we can't see, perhaps the machines can. And that this to, of course, machine learning. Okay, to get to our third topic, I'd like to briefly describe image registration. I think I'm a bit preaching to the choir. We all know that image registration is really critical in a lot of imaging experiments. Here's a case that Su Zhang has been using in our laboratory to look at glioblastoma patients using CEST MRI. CEST requires a series of images to be acquired over several minutes. I guess there it's 6 minutes and 24 seconds for total scan time for all those images. And therefore, if the patient moves, now that will destroy the quality of the data for doing the CEST MRI analysis. However, now with registration, you can see when the box turns green, this is with registration. The images can be registered to each other using pretty standard methods, and therefore the CEST MRI analysis method comes up pretty nicely, and you can see a percent CEST effect within the glioblastoma. If you don't do image registration, well, you just get a mess and very challenging then to analyze the results. So again, pretty obvious that image registration is needed when acquiring multiple images and then needing to ensure motion did not corrupt those images. I'd like to kind of extrapolate on that though and discuss multimodality imaging. In this case, MSOT and MRI. This is a great example from Sarah Bondiek and her colleagues in the UK, comparing MSOT and MRI, where MSOT is a multispectral optoacoustic tomography method where a mouse is basically placed in a plastic bag, suspended in a tank of water, irradiated with a laser, and then ultrasound signals come out of the mouse after being excited by this optical laser that's being shined into the mouse. So we won't deal with that technique in this presentation, but just to say you can get images, you can find tumors. Here are two subcutaneous tumors in the schematic, and here is the image. And then when the mouse was placed in the MRI, well, here are the same two tumors on that same mouse. And in fact, just to compare, I'm going to flip over this image for you. And right away, you can see that these two images do not compare very well. The mouse is relatively flattened in the MR image because it's been placed in this cradle that is relatively flat for the mouse. Now, while this baggy suspension system keeps the mouse in more of a cylindrical shape. In fact, if we were to show a schematic of that and then register, which is what the Bondic group did, you can see that the registration is not that great. And if you were to do this again, but in this case, the group built a silicone bed to hold the mouse in place, highlighted with this yellow flashing graphic here. Well, now you can see very clearly that the locations of the tumors match up better, and the positioning of the tumors, the shape of the tumors match up better. And again, if more schematically, as they did in their publication, you can see, yeah, this matches so much better than over here on the left, where you can hardly see that it is the same mouse. And the registration is much better. The dissimilarity coefficient is certainly much better. So I know this is about how to perform data analysis. That's the topic of our presentation. But again, to emphasize that you really have to think about how you're going to acquire the data in a good way so that you can simplify the analysis or make the analysis work for you. Otherwise, garbage in, garbage out, right? And I think this is somewhat obvious, but it's nice to see an example. So Here's another example of co-registration of MSOT and MRI, in this case done by Jan Kloos and his colleagues, where they were able to compare the MSOT and MRI of mouse brain, do some affine transformation in a rigid way, co-registration, and then also non-rigid registration can be applied in these cases as well. However, there are two important points that I want to make about this slide. The first is you got to curate your data. You got to make sure that your data is good quality before you go through some of these more advanced processes. And again, I like to call that data processing or image processing before you get to the true analysis step. And so in this case, they were denoising their data, very standard processing method. Then they can finally go through their more advanced analysis method to do the co-registration of the images. So it's a two-step or perhaps even a three-step process, right? Think of it as processing followed by analysis. And this is highlighted here too. Multi-step registration process improves spatial correlations. And the BoneD group found that as well. A whole body registration and then a tumor by tumor registration as a second step actually improved their results. So you got to think about this in multiple steps. In future directions, 3D registration, that's a little more automatic or through deep learning is an interesting idea as well. Finally, I just have a series of topics without any graphics. I know that's not the most compelling thing to see during a webinar, but these don't really lend themselves to graphics very well. I'd like to say just a few words about rigor in imaging studies. Of course, we like to say that we ensure our techniques work well before we start a study. We have good quality assurance. We know that an MR machine produces a relatively linear image along all three dimensions, so we can measure tumor volume relatively accurately, right? That's good assurance. You can think of other cases, a diffusion-weighted MRI. You might have a phantom, or you test the diffusion-weighted phantom and ensure that you're getting the right diffusion number before you actually start to study mice. However, I think what's underappreciated is that it's good to have quality control. And in the agriculture industry or the food industry, quality control is paramount. And samples are tested periodically to make sure that the entire crop or entire food line is safe, right? And we don't do that in biomedical imaging. It is very rare for us to add an extra mouse just to check for QA/QC or to repeat a study with a phantom to ensure that our equipment is working well. And I think it's something that's good to keep in mind. Certainly in genomics and proteomics, the same sample is slipped in with all of the other experimental samples as a QA/QC when a contract research organization is analyzing samples to ensure good quality control. And we should think about that as well to ensure that we have good data quality for our analysis. Also, I'd like to just briefly mention that we very often do repeatability studies. It is one example, we take one sample during one scan session, and we'll image it several times and get statistics and say, okay, we've got good repeatability, we have good rigor in our studies. And to be honest, that's usually pretty good. However, we should give some thought to reproducibility, multiple samples during multiple scan sessions during one day or perhaps multiple times on different days. This is challenging, and as we know, mice change over time. They are affected by the imaging process. If you were to knock me out for an hour and put me in a dark tube and then pull me out, I don't think I would be the same the rest of the afternoon, right? And it takes a little while to recover. And because of that, reproducibility studies are really challenging to think of carefully, and they're very costly as well. And yet, perhaps some consideration is needed now and then. I wanted to also then re-emphasize that this idea of gauge repeatability and reproducibility is a good statistics test for looking at the difference between the two. I'm a big fan of physiological monitoring and stabilization. Again, I think you need good quality data for good data fitting. And I work very hard to make sure that the body temperature of mice is exactly at 37 degrees, plus or minus zero point 0.2 to zero point 0.5 degrees. I'll live with zero point 0.5. Anything above zero point 0.5, and I think really you got to work at the temperature. Again, that depends on your study. So if you're measuring tumor size, not so critical. For the CEST MRI studies that I do, measuring chemical exchange rates, yes, temperature is important. And also respiration rate. I think we should have a good physiological consistency among our mice. And if you have highly variable mice in your study, well, it just stands that there's going to be more variability in your analysis at the end as well, right? And one way to check for consistency among your mice is the respiration rate. Of course, you could also look for cardiac rate as well, but I think the respiration rate is really the easiest to control, along with the body temperature. So really critical to have good control. In fact, we've gone as far as to buy these physiological monitoring systems from a company and put it onto other instrumentation just to have a quality control periodically during a study to ensure that our mice are always at 37 degrees and we always check the respiration rate. On occasion, we have sheets where the operator needs to fill it out every 15 minutes to record the respiration rate as a way of ensuring that we have good quality. And finally, in the time remaining here, just a few minutes, um, I just wanted to describe that there are three levels of imaging productivity. We're researchers in the field of MRI, and therefore we think of our research investigations. And then there are facilities who perform these methods, small animal imaging facilities, for other researchers who are not experts in MRI. Well, I think really there's an intermediate level of advanced research service that we're getting to with diffusion weighted MRI, DC MRI. I mentioned the CEST MRI, I think would fall in that category. Someday I think hyperpolarized C13 MRI might make it from research investigation to advanced research service. And the key, and this is the important point, is the image analysis that defines the difference between a facility service and advanced research service. If you need higher end image analysis, then you need to go to advanced research services to get the job done. And a lot of our facilities are not set up that way. A lot of our research groups are not also set up to help others with their advanced analysis. And this is a problem in the field that needs to be solved, and I don't really have a good solution. And I think at that, it is a great point to end this presentation so that we can get to the Q&A where I will look forward to all of your provocative questions about advanced research services. That I'd like to thank all the people in my research group. This is what we look like when we're in the laboratory or when we are robbing a bank. And this is actually what we look like when we're at a virtual meeting. And I can't thank them enough for everything they do, along with our collaborators and our funding sources. And with that, I'd like to thank you for your time.